Welcome, Bio4. Welcome to the third part of our spinal nerves. So what we're going to move on to now are the spinal meninges. And the meninges are the coverings, the coatings that we have both in the spine and in the brain. So let's take a look at those. They're specialized, and what they're going to do is going to provide some stability and shock absorptions. Just think of it, I always think of it as a duct tape wrapper around something that's just going to hold things in place. And these meninges or layers are three of them. So we're going to look at them independently. The outermost, farthest from the brain here, and the outermost in the spine also, they've given it the name of the hard mother. It's a little weird. And it's called the Dura Mater. So Dura Mater is dense connective tissue. It's something that if you wanted to rip it with your hands, you absolutely could not. It's very, very stiff and a real duct tape of a coating here, of a covering. The next one is called Arachnoid Membrane. And arachnoid is from spider, of course, and it does look like a spider web. So it's these fine little fibers, very easy to tear, fine fibers that are all arranged in a second layer sort of immediately below the Dura Mater, and then a third layer called the Pia Mater, or delicate mother. Which really, when we do our dissections of the brain, we can't see that. It's one layer of cells. It's very attached onto the brain itself, and it's not something you'd see. But you can see the Dura Mater and the arachnoid membranes, and sort of peel them off the brain when we dissect in person. In lab, we do sheep brains. Okay, so let's look at the innermost first, the Pia Mater. It's really firmly bound to those cells that make up the brain, and it contains some of those blood vessels that are going to supply either the brain or the spinal cord. So it's loaded with little vessels that are actually feeding into this really important tissue. The arachnoid then is simple squamous epithelium. You had a squamous epithelium in your tissues, and it's a network of fibers. So these fibers are both collagen and elastin, and they form a little layer, and then they send like pilings down on to the Pia Mater. I'll show you pictures of that. See, so this would be the arachnoid membrane. This would be the surface of the brain here with the gray matter and white matter. And the Pia would be closely attached onto the brain, and then with these pilings coming up like this is the arachnoid membrane. So this kind of looks like spider webby, hence the name. This enlargement here, it helps you, the brain, and then this little space, and then the arachnoid membrane. Now, important is the space that it leaves by the pilings. So internal to that membrane then is that space, and what flows there is cerebrospinal fluid. Remember, we talked about that before. It's a big shock absorber. It's also a very good place to float gases and nutrients and wastes. Okay, so let's go back to the picture again. You're going to see floating all around here is cerebrospinal fluid. If a spinal tap has to be done, where you have to examine the cerebrospinal fluid, this is what happens. They have to go in with a syringe and go past the Dura Mater and actually go under the arachnoid space and collect some of that. This is a case of wanting to see if there is an inflammation of the meninges, right? We're talking about meninges, and that inflammation of the meninges is meningitis. So it's usually to test for meningitis. I know that individuals who do have this done have horrific headaches, I think from the drop in pressure of that fluid. The Dura Mater is the tough, fibrous outermost layer, and it covers not only the brain, it covers the spinal cord too. And you've heard some of these terms. For superficial to the Dura Mater is the epidural space. Okay, superficial. It doesn't, the epidural space does not, you don't have a needle going down through the dura mater. It's superficial to it. And deep to the dura mater would be the subdural space. Okay. This dura mater covers the brain, and it also divides the right and left section, so it sort of flows into the center of the brain. The correct term is to invaginate. It comes into the center and sort of separates both hemispheres. So thinking more about the epidural anesthesia, it's given in that space. It's a space inside the bony spinal canal, but superficial to the dura mater. So let's look at a picture. So if you had the spinal cord and you had spinal fluid and you're coming in between the spinous processes here, the catheter has to come in here, and it won't come down into the spinal fluid, but just in this region here, in that epidural space, is where that is deposited. In a different view here, if you're coming in with your needle, you are not piercing any of this blue dura mater here. It's coming in, and in this fatty region here is where it's deposited. It's very interesting because you're not exactly sure which way it's going to flow. So a little will, if you're lying down, a little bit will go, cranially, a little bit will flow in either direction, but you're not certain how much. It's just kind of like a little bit of a shot in the dark. So let's move on now to the spinal nerves. And this is a model. It's kind of nice to look at these of the vertebral column because you get an idea of those exiting spinal nerves. So we've left the cord now and we're exiting, through that space, right? Between two vertebrae, remember, intervertebral space. And these nerves are exiting. And remember, there was an enlargement, right? There was a cervical plexus here, and then there's going to be a brachial plexus. Then it looks pretty straight. Look, see this tanglement, these tangled nerves are called a plexus. So we're going to look at that more closely, but just for now, get sort of a quick idea on, oh yeah, neat and tidy, oh messy, oh yeah, neat and tidy, oh yeah, messy. Okay? All right. So what's a nerve again? A collection of neurons in the peripheral nervous system. So if this big thing were a nerve, right, then we're going to have these clusters called a fascicle. 2, 3, 4, 5, 6, 7. Was it 2, 3, 4, 5, 6, 7, 8 fascicles in this particular nerve? And within each one of those fascicles would be an axon. In this case, you're not seeing the cell body; you're just seeing the axon that's myelinated. So the coating around here, around a particular nerve, is called the epineurum when it comes around the entire nerve. So I'm directing you to here, epineurum. Perineurum is this coating that exists around a fascicle. And then an endoneurum is the coating that exists around one particular neuron. Okay, epi, peri, and endo. I'm going to make this the last slide. So we have spinal nerves. We have 31 spinal nerves, each named by the vertebrae in the proximity. So we're going to have 8 cervical, 12 thoracic, 5 lumbar, 5 sacral, and 1 coccygeal. This should remind you a little bit when we were talking about the bones, but we had breakfast at 7. We didn't have breakfast at 8. So why is this different with spinal nerves? Just think that, taking you back, that there is one nerve above your C1 and one below it. So you're already adding an extra one because there's going to be an extra one below your last 7th cervical vertebra. So you're adding an extra nerve because It's both above and below that first bone. Okay, let's stop here and we'll continue on with the spinal nerves in the next section. Thank you.